Hi, and welcome to PLIST. PLIST stands for Powerlink Lucid Identification Support Tool. PLIST is a multimedia identification tool used to identify or diagnose unknown weed and plant species. PLIST now covers all plants of interest, including weeds, rare and endangered plants, and is a repository for all plant resources for Powerlink Queensland. The aim of this tool is to enable Powerlink and their contractors to identify weeds, undesirable species, and other plants of significance that may be found growing along power line corridors and/or on other Powerlink managed properties in Queensland. Identities. When you open PLIST identification key, 4 windows are displayed. The top left window contains characters and their states. The plus and minus signs beside the characters allow you to expand and compact the available options. Select the states by clicking the thumbnail images, or when thumbnails are not showing, in the checkbox on the left side of the featured text. In the top right-hand window is a list of all the species that are contained within the key. As you select features and states, some species will be discarded from this list. The discarded species are displayed in the lower right window. The aim is to reduce the number of possible species in the top right window to 1 or just a few. Once you have identified the species in question, you can click on the attached fact sheet, which will provide you with comprehensive identity information. Now let's go through the process of using PLIST. When trying to identify a specimen, you can choose any feature or state in its list at any time. But stepping through the key in a structured and sensible way will make the task of identification easier. If you are also familiar with PLIST, then you may already know many of the specimen characteristics. Briefly reviewing these before you start will make it easier for you to proceed through the identification. As you can see here, features include plant, stem, leaf, flower, etc. characters. Expand the feature to see the subsets such as flower colour and size. It is also important to be familiar with the characteristics of the specimen you wish to identify. Take for example this plant or weed here. Note and use distinctive features of your plant or weed. In any key, some taxa, or group of organisms, may possess particularly distinctive features. Use of these may allow the specific entity to be keyed out in very few steps. Selecting character states for the most distinctive features may quickly reduce the list of entities remaining. In this instance, the most obvious feature is the blue flower, but also take note of the petal and leaf characteristics. Consider the characteristics that were listed in PLIST. So, if we select flower colour as being blue, we can see now that we have 190 remaining entities, with 1,560 being discarded. Remember, you don't have to make a selection from each feature, and it doesn't matter what order you select the features in. So, another obvious feature is petals or rays. Note, always choose multiple states, that is, more than one state or feature, if you are uncertain which state is the correct one to choose for a particular specimen. PLIST is designed to allow you to choose as many states as you require from any one feature. Within the program's logic, these states will be connected by an OR link. This will cause PLIST to search for all taxa or organisms with any of the states you select. As a general rule, if you are unsure which of two or more states your specimen has, then choose both. That way, you can be sure your target taxon will remain in the entities remaining. For instance, if we thought this flower looked like it had 2 petals, when in actual fact it had 3, you would eliminate the entity you are interested in. So to be safe, select 1 or 2 and 3. The remaining entities won't reduce as much, but at least you won't have accidentally eliminated the entity you are looking for because you had an incorrect assessment of the number of petals. So as you can see, we are now down to 49 remaining entities. Note in the lower left-hand window is a list of all the features chosen so far. Let's look at another obvious feature, the leaf. As you get more familiar with identifying plants, paying attention to leaf size, arrangement, and attachment will become second nature. Consider things such as size and shape. Taking a photo of a leaf against a hand will help gauge the size more accurately in order to identify later. The largest leaves fall around the 5 centimetre mark, whereas the majority actually fall under that. So it's safe to say small in this case. We have now reduced our number of possibilities to 33. Now, from this close-up image, we can see that the leaf actually has hairs. Let's scroll down to the characteristic and choose present. We now have reduced the possibilities to 25. When you have dealt with all the obvious features, use PLIST's best function to suggest the best remaining features. Clicking on the best button will move to and open the best available feature. If there is difficulty in addressing the first feature nominated, next best and previous best buttons allow you to navigate through the feature list. In this instance, best suggests fruit type. Since there is no evidence of fruit, use next best, and the option is district. In this case, the sample was growing at Morton. We now have 7 remaining entities. Another tool called Prune Redundant Features will remove features that no longer remove any species from the remaining list. This makes the identification process far more efficient. Many users find it useful to use the 2 buttons, Best and Prune, alternately, one after the other. Or alternatively, you can automate these features via the Feature Automate menu option. So let's click on Prune now, and then click on Next Best again. The suggestion is plant form. These thumbnails help you in identifying the form. However, if you are still unsure, there is a text description that can help. The specimen we are trying to identify looks like a vine. When we click on the vine, we are left with one entity. Click on the fact sheet icon to view comprehensive entity information, including images. Look through all the images to see if we have made a correct identification. Note that some plants and weeds can vary in appearance due to age or time of year. In this instance, these images do not match our specimen. So let's look at the last selection made. We selected vine. Click on the text description icon to read its description. The most obvious feature of a vine is that it has tendrils, a coiling stem that attaches a plant to its support. If you look at our specimen again, you will notice there are no tendrils. Since this was an incorrect selection, we can deselect it by clicking on the thumbnail. As you can see in the Features Chosen window, the vine thumbnail is greyed out. And we are back to 7 remaining entities. Since we are unsure of plant form, let's move on using next best again. This time it's leaf arrangement. Check your specimen and make your selection. In this case, alternate. We now have 4 remaining entities. Never assume that you will always end up with one entity species remaining. Some species in the key may be very hard to differentiate, except when using difficult or obscure features. Sometimes, after you have addressed all the features, you may have a short list of species remaining instead of just one, such as the case here. In this instance, we could carefully check the specimen against the information provided in the fact sheet. Alternatively, we could try the Differences tool near the right-hand end of the toolbar. This gives a list of all the features and identifies the differences for that specific feature for each of the remaining entities. Take, for example, flower type. 3 have tubular flowers, which helps to single out our one specimen since it did not have a tubular flower. Close this window and read the attached fact sheet, which will provide you with comprehensive entity information and images, which indeed confirms that we have identified the specimen correctly. What if there are no entities remaining? This will happen sooner or later in one of your identification sessions. If no species are listed in the Entities Remaining window, then it simply means that no species in the database match the selection of states you have made. Several explanations are possible. The most likely error is that you have made an error in one or more states that you selected. The species may also be undescribed. In this case, the specimen cannot be identified because its features are not represented in the key's data tables. Lastly, the key author may have made an error when constructing the key. This is unlikely, but it can happen. If, after carefully checking all the features and states, and checking that the specimen you are attempting to identify would be expected to be included in the key, then a key construction error may be present. Whichever situation is suspected, you must very carefully review your chosen features and determine which ones you are uncertain about. Try unselecting uncertain states one by one to see what effect each has. One or more species may move back to the Entities Remaining window. In difficult cases, you may need to play with the key, adding or deleting states progressively to try to find the best matching species. Once you have made a preliminary identification, check the other information, notes, description, and images provided for the species. Getting a possible name for a species from a key is not the end of an identification. You need to carefully review the associated information to get a good indication as to whether the resultant species is correct.